Okay, so welcome everyone to today's edition of Tissue Talks. Um, we have a really special speaker today. So this is Kelly Stevens from University of Washington in Seattle. Kelly is one of our speakers that gave a talk before, like a few years ago. I think it was still a COVID time. And her talk was really well received. And so we occasionally invite our early speakers back to tell us what all happened in their science and also in the broader field. Kelly is Kelly trained in biomedical engineering at the University of Wisconsin-Madison. He graduated in 2002 to move to the University of Washington to obtain her PhD and continue with the postdoctoral training at MIT from 2009 to 2014. She has a very broad spectrum of research interests that are actually very nicely coherent. And to just quote her own words about what her research is, she says, our research seeks to hijack and rewire aspects of nature's developmental programs to control the processes by which cells assemble to form human tissues. To do this, we use diverse tools taken from stem cell biology, tissue engineering, synthetic biology, micro and nanofabrication, and bioprinting. So this broad spectrum of areas really focuses on a couple sort of practical areas of application, which are infectious diseases, development, regeneration of the organs. And over the years, Kelly mostly worked in the context of heart and liver diseases. She also says something very interesting on her website, which is explanation about how she is inventing the future of medicine. That this happens by developing technologies to assemble human tissues from stem cells and to remotely control these tissues after implantation into the patient. In addition to her scientific research, Kelly has been invested, I think, throughout her career into fighting different types of inequality. And we still remember her very nice article in Nature Reviews Materials about how to how to minimize the disparities in visibility of scientists and their research. Along these lines, Kelly is now a co-PI of a new center that is funded by the National Institutes of Health and with a mission to reduce disparities in biomaterials research. This center is advancing biomaterials research and clinical applications while connecting researchers at well-researched institutions with the researchers that are in institutions that offer really diverse talent rather than diversity of resources. Her work is very visible, and she was recognized and awarded in multiple ways. It all started during her training when she was a graduate research fellow of the National Science Foundation. She also got a fellowship from the NIH. She got a Wellcome Trust Image Award, and she was one of the new voices in the cohort assembled with national academies. Finally, Kelly was elected to AIMBE, American Institute of Medical Biological Engineering, and last year she also obtained the Mid-Career Award from the Biomedical Engineering Society. Kelly, thank you so much for being with us here today, and you can now take over the control of the screen.
Thank you, Gordana. That was quite the intro. I'm honored. Thank you so much. It's always a delight to get to be introduced by one of your heroes, so thank you. And I should say, I'm impressed by the attendance at this tissue talk. This is a seminar series now, you all know. It's been going on for a few years, basically since Gordana and her colleagues at Columbia were visionary enough to realize that it was needed during COVID. And the fact that we're still having such great participation, even many years later, is just awesome. And I just want to say I'm happy to be a part of it. I'm going to tell you a little bit about our work in my lab, as well as some of the work that I've been doing with colleagues across our profession to build human organs. And specifically today, I'm going to focus on most of our work in the field of how can we bioprint a liver, okay? And Gordana made it very clear that I need to show you guys some data that I didn't show last time. And I wanted to give a shout out to our colleagues in New York, again, for hosting this awesome seminar series.
Kelly, I think, can you reshare your screen?
Yep, you got it, Naveed. Thank you. Okay. Did we lose it?
Yeah, try again.
All right.
Perfect.
Yeah.
That worked better? Okay. I want to give a shout out to our colleagues in New York City. So for those of you that I was actually pretty impressed right when I came on the call that both Gordana and Naveed recognized the statue right off the bat since it's in their hometown. This is Prometheus, who stole the eternal flame or fire from the Greek gods. And the reason that I'm showing it to you is because it's a symbol of Prometheus is often thought in the field of engineering as being a symbol of advancement and thoughtfulness, because what he did was he gave humankind, at the time humans were this marginalized population, he empowered them and allowed them to innovate, right? And so I just really think that that's amazing. In concept, how do we bring people in, or bring beings in to innovate in our profession? And the other reason that I want to show you guys this is because it's also the first time in known human history that folks talked about the power of the liver to regenerate. So I'm going to this is kind of gross, so I'm going to be quick.
Kelly, can you share your screen? I mean, you need to go to presentation mode.
It's not working still, huh?
Can you just reshare?
Now it is. Yeah, yeah. Okay.
Is it going?
Yeah.
Just go to reshare your screen. So like cancel the share and then.
Stopping sharing.
Hmm.
Let me share it again.
Awesome.
We're flipping again now?
Yep, no, it's perfect. Thank you.
You know, so the thing with, so Prometheus, obviously the Greek gods did not approve of the fact that he stole fire from them and gave it to humans. And so to punish him, they chained him to a rock where where an eagle ate his liver, and it regenerated over and over and over again. And so that's kind of a gross story, but it also tells you that even several thousand years ago, humans had figured out that the liver is actually the only visceral organ in our body that can fully regenerate if you cut a piece out. Okay. And so imagine if a surgeon were to go in and surgically resect up to 2/3 of your liver, you could actually entirely grow back that liver within just a couple of weeks, assuming that the liver doesn't have any underlying injury. Okay. Unfortunately, that's not the case if there's an underlying injury that has accumulated over time or acutely, okay? And so I saw that there's about a little over 100 folks on the call today, and I would bet that many of you guys are students or trainees, right, in the age group of, say, 20 to 30. The leading cause of hospitalization for folks in your age group is what's called acute liver failure. And that's basically if you take some kind of a substance. The most common one that we hear about is Tylenol, but honestly, there's a bunch more. If you take a substance, let's say that you went out partying for a night and then you came home and took too much medication afterwards, okay? And basically you're taking a couple of different insults on top of one another. What will happen is you can send your liver into what's called acute liver failure. And unfortunately, it's a huge cause of transplant. It's a huge cause of mortality. And one of the things that we're wondering in my group is, well, what if we could help a patient by giving them an artificial liver just for a couple of weeks, okay? Because what's been shown by clinicians is if you can get your liver to last for just a week or two, it can actually sort of get over that chronic, or I mean that acute damage, okay? And it will actually be able to regenerate itself, okay? So if you can give a patient a transient, so short-term bioprinted organ, even for a couple of weeks, you would then be able to take that organ back out and the patient's liver would be fully regenerated, okay? So we think of this as an actual tangible way to actually bring bioprinted organs into the clinic in the short term. You can imagine that doing this longer term, like trying to sustain a patient for the rest of their lives on a bioprinted organ, that might be a little bit tough, right? But we're thinking that this particular avenue is a way to get this into these type of technologies into the clinic sooner than later, maybe even within the next few years, right? So when my lab first started working on this, we said, how do we build human livers? And we started in the lower left corner saying things like 3D printing and microfabrication and all those technologies that Gordana had mentioned earlier. We quickly realized that we really don't understand what human organs look like at the cell level. We simply don't have the blueprints or the maps that we need to build these organs, okay? And so let me give you an example. So this is, if you were going to look in a standard textbook, what a human liver looks like, this is what you might see, okay? So in the upper left corner, you've got a zoomed in view. So the liver looks boring macrostructurally, right? If you see on the upper left, it looks like a big brown blob. You zoom into that, and what you're going to start to see at the microstructural level, so if you pulled out a microscope, are what's called the hepatic lobules, okay? These are repeating approximately hexagonal units, and those are called the functional units of the liver, okay? And within each functional unit or lobule, you'll see hepatocytes. Those are the functional cells. They perform hundreds of chemical processing. So they're like the chemical engineers of the liver, and they do everything from making proteins to metabolizing drugs, okay? So these are the cells in sort of a peach color here. And they're so metabolically active. They need so much fuel to do what they do that they're really, really richly supplied by blood, okay? And so there's a couple of different vascular networks that feed blood into the liver. So the blood comes in through the portal network out here on the external of the lobule, and then it crosses through the hepatic sinusoids, and then it goes out, dumps out through this big central vein. So there's a couple of major vascular networks that surround the lobules and then also go down the axis of them. And then there's also finally a bile duct network that basically takes waste out of the liver, and that's in green here. Okay, so just imagine you've got this really extravagant network of tubes that surround the lobules to give them blood and then also to take waste away, right? Now, again, this is an artist rendition of what this looks like, and a lot of the way that these images were drawn, frankly, is like, this is not reality, right? This is what folks think it might look like based on 100 or so years of histological sections. So this is when, you take a razor blade and you slice the liver and then do a stain on it, and then you can see things like the hepatocytes here. Okay, these are the cells in the pink, and then these are the sinusoids or the capillaries that go throughout, okay? And so really the question is, how do we now take these images that we have and use a 3D printer? So if you were a student in my lab and I said, great, now go 3D print a liver, you know that this one's not accurate, the artistic rendition, and you know that the histological one, I mean, how do you make that into 3D, right? So it's actually not a trivial concept. And so one of the things that we've been doing is working on tissue clearing and immunostaining and other advances to start to get better 3D looks at what some of these structures look like, okay? So here I'm showing you the major blood vessels that surround one of those lobules. So this is a lobule right here, and this is two different of the vascular networks, one in orange and one in purple, the portal triad. And then here you're seeing the bile ducts that empty waste out of the liver. And cyan, and I hope what you can appreciate already is just frankly how complex it is, even in just this small little section. And then also how these networks are entangled and wrap around one another, okay? We've been able now to do this across scales. So here we're looking at a section, it's only still a few millimeters across. By the way, we're not anywhere close to getting a full liver, but here you can see each lobule The center of the lobule, the functional unit, is in yellow. And the exterior of the lobule is mostly in the cyan colors. And you zoom in and remember when I showed you the sinusoids, these are the blood vessels in the liver in this artistic drawing. Look at how they kind of look like spokes on a wheel here in the art drawing. All right, now let's look at what they actually look like. Here are the sinusoids, so these blood vessels that connect across the lobule. And what I hope you can appreciate is how much more tortuous and complex they are. Here is an even higher magnification view, and it's almost more like a net that comes across the lobules, right? These are the biocanaliculi that again take waste out. Each hepatocyte cell is sort of in each of these little black areas here. So it's vastly complex. We're not even close to recapturing the complexity of these organs when we are doing the artistic drawings and the histology. We just can't even comprehend, right? And so I just want to encourage us as bioengineers, especially those doing tissue engineering, to really start to, when you're building your organs, not just go off of an artistic rendering, right? But really to start to question, is that accurate, and what do these really look like? Some of the things that we're starting to notice now is, for example, the different vascular networks in the organ have really different topologies. So this is the central vein network. This is the portal vein network. And what we're starting to do quantifications on these now, and so showing, for example, they have different branch angles and different diameters, and even the different vascular networks within the same organ that are entangled have different vascular topologies, right? And then, of course, as you cross across different organs, it's even more different. And so at the moment, the tissue engineering field is just really not capturing these differences in architecture at all. As you'll see actually in the rest of my talk, my lab is still not even doing it. And so I just want to draw our attention to this and start thinking about it. This is, again, this is a larger network, right? And here is the vascular topology of the sinusoids that was captured really recently by Doris Gonnet in the lab. And what I hope you can, again, appreciate is just the vastly different looks in architectures and topologies of these organs. So stay tuned as we're doing more quantification and thinking about how to algorithmically model that and everything in future. Okay, so I'm going to skip then to this next part, which is, as we start to map these organs, then how do we build it? I think that's probably what most of y'all are most interested in, this being a tissue engineering audience. I think many folks in this field over the last few years now have started to think about, okay, well, let's start with the building blocks that we find in the organs. And most of us would first think cells, or many of us, right? And in this case, with the liver, the vast majority of the cells in the liver, and for sure responsible for most of the liver's mass and function, are the hepatocytes. And so we've been working in my group over the last few years to try and isolate hepatocytes from humans and to then propagate them in vitro. And that's actually a challenge that has been incredibly, although, we've known that livers can regenerate in humans and in animals since Prometheus. Interestingly, we haven't been able to get those hepatocytes to multiply and, quote, regenerate in a dish until literally about 6 months ago. And so there's been some really, really big breakthroughs in this field. This has been what folks have thought of as an Achilles heel in this field for decades and decades and decades. And finally, we're closer to now taking primary hepatocytes from a patient or an organ donor and then culturing them, in this case, as organoids. So like many of you, we put them into Matrigel and then propagate them as organoids, right? And what we find is that these organoids they express markers associated with hepatocytes here, cytoskeletal proteins, cell junctional proteins, functional proteins like arginase 1 that's involved in nitrogen metabolism, transcription factors like HNF4 alpha associated with hepatocytes. So just, I think you're going to see in the liver tissue engineering field anyway, a lot of progress in the next, say, 5 or 10 years, as finally we have a more expandable cell source. I want to draw your attention to the scale bars on these, okay? Again, something that our field tends to minimize is, okay, so we can culture organoids in vitro now. Yay, that's great, right? That's huge, again, huge breakthrough. Unfortunately, the problem with this is that organoid in general is 50, 100, maybe a couple hundred microns across. Okay, that is very, very tiny. And I like to start to give a metaphor for how tiny that is, okay? So that you can understand the scaling challenge we're starting to think about. If an organoid is the size of a mouse, okay, so just one organoid in your cultures, I know many of you culturing organoids right now. So organoid is the size of a mouse. A human organ is going to be more like 65 elephants. And in fact, a human liver is going to be more like 700 elephants, okay? And so it's pretty mind-boggling how far off in scale we are. So we can culture organoids, but we cannot create human organs yet. And the scaling that's really required to make that leap is going to be a really big challenge for our field. Many of us now are starting to try and mimic the way that biology does that by building a vascular tree, right, to scale organs. I'm going to start with some work that we did with Jordan Miller's lab when he was then at Rice University. And this is a 3D printing method called digital light projection. Okay, so now we're saying, can we bioprint human organs? Okay, and in order to build these vascular networks within them. So imagine you have a liquid that is light sensitive, okay? And so you can put your liquid in a bath that's in yellow, and then you can flash light on it like the projector, okay? And as you're flashing the light, you can change the patterns that you're projecting, okay? So over here in the lower left, and if you move your tissue that's emerging in the Z direction, what can happen is you'll essentially get this engineered organ to rise out of the bath. Okay, you've probably seen videos of people doing this kind of printing with like the Eiffel Tower rising out of the bath. So here's what it looks like. You flash your different photomasks in the upper left, and your engineered organ rises out of the bath in the right. Okay. And the reason that we liked this technology at the time, we still do, is because we can make these bioprinted structures out of hydrogel. So that is aqueous materials, right, that are hospitable to human cells. And we can make entangled networks in them. What we're trying to demonstrate here really is just that these networks are entangled, similar to how they are in biology, right? So we've started the lab with that technology, and I'm going to actually switch gears talking about it because we were excited about the entangled topologies, but one of the things that is an Achilles heel of this, and part of the reason I'm even showing this, frankly, is just because it gets in a flashy paper doesn't mean that it's perfect, right? And I think it's really important for our field to start thinking about when we're picking a 3D printing technology, how are the cells being exposed? What conditions are the cells being exposed to while that tissue is being constructed? Okay. And so let me give you an example here. And just like in our digital light projection that I just showed you, in this schematic, what you see, this is a schematic of an extrusion printer that Sharina Malkani and Liv Prado in my lab made. And basically what you see is as the printing is happening, your living human cells in blue are just sitting here, okay? They don't have access to any kind of blood supply then. They don't have access to any nutrients, and they're just sitting in that bath. And they're also exposed to printing conditions sometimes, which are bad, which are also deleterious to the cells, okay? And so this is true of a lot of different extrusion techniques. It's also true of digital light projection where they're just sitting in that bath. And often this printing process, and this is something we don't often talk about, takes a minimum of hours. And I'm talking minimum. And in some of the more organ scale work that's been done, I mean, Adam Feinberg has some really cool stuff, right, where they've printed a whole organ scale human heart. That one took 2 days to print, right? And so you can imagine if your cells are present during the printing, they're dead by the end of it, right? And so one of the things that we and other folks in the field have been working to say is, well, what if we can decouple the printing from when the cells are present, right? So what if we can instead make what we call a sacrificial template? So now we 3D print a structure, Okay, and there's no cells present during that 3D printed structure. It's just this freestanding thing. And then we can encase that structure within cellularized matrix. Okay, so now we're going to add in the cells in the hydrogel. And then all you have to do is flush out the structure, and now you're left with your printed topology. Okay, so this is called sacrificial templating. It's not just our group that's been doing this. There's some fabulous recent work by Chris Chen's group in this area, and Jennifer Lewis has got some nice, Mark Scott. I mean, there's a whole bunch of folks that are doing really nice work in this space, okay? The idea here is print before and then add the cells, okay? And then print out your your structure. We've done this with a couple of different ways of printing, which I'm going to actually bring up again later. But again, here's how it works. You're going to print your sacrificial topologies. Then you're going to collect your templates. Then you're going to encase them in a hydrogel, evacuate out the template. And then if you want, you can line your channels with endothelial cells, right? And then you have your engineered tissue out the other end. So again, you print your template. You can encapsulate it in a biomaterial containing cells. This is actually us doing this now, right? Evacuate out the template, and then you can perfuse it, okay? And what we found is that by doing it that way, this is an example of 3D printing sugar, okay? So imagine you have these really fancy desserts that you get in a really fancy restaurant, and it has like patterned sugar lattices, right? It's kind of similar, actually. So this is laser sintered carbohydrates that you can make a structure like this, and then you cast cellular cells around it here. So you can cast a biomaterial containing primary hepatocytes around it. And here, each little dot that you're seeing, and down here, each little dot that you're seeing is actually a cellular aggregate of hepatocytes. So if you zoom into that, a higher magnification, you're going to see each single hepatocyte here is its nucleus and each single cell, okay? So the cross section is we're able to make these sort of coarse vascular networks throughout these tissues. One of the reasons the field was excited about this at the time is because this tissue was about 3 centimeters, a little less than 3 centimeters in length, okay, which folks thought of as huge, okay, at the time. And so again, in terms of scaling, this type of a technology is allowing us to get closer, right? However, we still have a ways to go. And not only that, but I always like to, again, show areas that we can improve. And what I want to point out here is if you look at sort of a middle magnification view of this organ, what you can see is that there's huge gaps between the cells. Okay, so this is a hepatocyte aggregate, this is a hepatocyte aggregate, and there's huge gaps. I mean, if you look a really zoomed out view, you sort of don't see that as much, right? So I encourage you all to look at all of the data. But this is not how densely cellularized, this is not even close to capturing the dense cellularization that we saw when we were doing those liver map studies earlier, right? And so one of the reasons why is because the biomaterials that we're using in these cases are not amenable to letting the cells remodel and grow within the structures. And really, this has been a big gap in the field of how do we take these organoids and put them into printable bioinks that support cellularization, integrate well with the host, become richly vascularized. And as y'all know that are doing this is just really tedious. And we start to look at what material to use and what cell population to use. It takes forever to try and work through these studies, especially once we start to work to implant these within an animal host, right? So you might find something that works okay in vitro and your cells produce some functions, but then you implant it in vivo and the tissue either dies or it doesn't integrate or whatever, right? And so one of the things, and this is something that I briefly presented last time I was giving a tissue talk when it was unpublished and sort of sloppy and not hashed out. And now we have a much more refined version that I'll give you a little bit on before moving on to some of the newer unpublished stuff. But the idea here is it used to be that we would take, a biomaterial and then transplant, add some cells into it and transplant this into a mouse, right? And we said, well, why can't we instead take a 3D printed well plate using some of our hydrogels with a 3D printer and put different materials and different cell combinations in and transplant them all into the well plate, right? Which we can then transplant into the animal. Okay, so this basically allows us to look at all of the different printable materials and cell combinations at once, right? And so this is how this system works. We can make different printable well plates. Think of it as a hydrogel well plate. And you can fill them with dyes in this case, or you can fill them with cells. And here we just did a super early proof of concept study, okay, where we said, well, what if we just fill it with all the same cell formulation to start with? So these are going to be endothelial cells, okay? HUVECs, and then we put them in different materials. Okay, so we're just going to very nuanced, make very small change of different weight percent of gelatin methacrylate. Same cell combination goes across the array in this first study. Then you can implant the whole array, and then you can explant the array after a week or two and do the 3D imaging on it using the liver map or the mapping work that we did earlier, the 3D immunostaining, and analyze it. So when you explant the array, this is what it looks like. On the left, it's basically a jello array, and it's got animal fat apposed over the top of it. And then do the 3D immunostaining on it for human endothelial cells in red, and green is mouse erythrocytes, so blood cells, okay? So what that means is any well that has robust green networks in it is a network that has connected, that has formed, is lined with human endothelial cells and contains mouse blood. So that means that these human endothelial cells have formed a network that's been able to connect with the rodent vasculature to carry blood, right? So in other words, here, yellow is good, so to speak, right? And what I want to point out is these materials are all the same. They're basically the 3 and 5%, so the softer materials, okay? And when we then unblind the entire array, you're able to see a nice dose response here with the gelatin methacrylate, where the lower weight percents of the gelatin methacrylate much more robustly support that vascular network formation. Okay, so in other words, we can really, first of all, something as finite as just very nuanced changes in weight percent of a material will have a pretty major outcome on, in this case, vascular self-assembly and inoculation with a host, right? And then more importantly, we've been able to screen these all within one animal, right? Okay, so we then went on and did many more conditions within the array. I'm not going to dwell on this since it's now published, but the basic finding here is that we were able to identify combinations that are both supporting cell assembly and 3D printable, okay? And so one of the, again, the challenge has been that just because we understand what's good for cell assembly doesn't mean that that material is robustly 3D printable, right? In the past, materials that have been stiffer, like up here, are easier to print, whereas materials that down here are sort of more amenable to cellular organization, right? And so now we're starting to be able to identify materials that are sort of the Goldilocks materials that are good for both, okay? And we hope that that will help us as the field moves forward. And importantly, one of the reasons this has been so helpful for the field and for our lab as we're trying to identify new formulations is we used to do a study with 126 mice in 1.5 years, and now that same study takes 8 mice in a month, okay? We've been able to get these hepatocytes. We've been able to identify conditions that also support hepatocytes in these gels. Okay, so here I'm showing hepatocytes in pink in the upper left, and then the vascular networks in cyan. You can see the hepatocyte aggregates in the networks. And then interestingly, and actually this is in line with some of the stuff that we've seen previously in prior work, is that when we then elicit liver injury in the animal, these hepatocytes undergo massive remodeling and actually start to take on really, really architectures that are very much similar to the liver's architecture. Okay, so we hope then that we're getting closer to being able to identify materials that are 3D printable, and also that the screening technology will bridge that gap. So then the idea is, okay, well, now we've identified materials that better support vascular self-assembly. And the reviewers of our papers, and for good reason, said, well, do you even need 3D printing? Right? I mean, if the cells can just assemble on their own, you shouldn't actually even need 3D printing at all, right? If you have the right material. And we said, well, that's a good thought, but the problem is if you take a big gel and you put your endothelial cells and fibroblasts in it and then transplant that into an animal, and here, when I say big gel, I mean basically as big as you can get in a mouse, which is, in this case, it is about 1 centimeter across or a little bit less, okay? And you transplant that into the animal, and then you do your 3D immunostaining after about a week. And what you'll see again is when you stain for human endothelial cells and mouse blood in blue and human endothelial cells in magenta, what you see is that yes, the mouse vessels will reach in to connect with the human self-assembled vessels, okay? And they will perfuse that tissue um, up to a certain distance, up to about a couple of 100 microns into the gel. And then all of a sudden what you get are some human endothelial cells that are trying to self-assemble, but can't get access to that blood. Okay, and so in other words, yep, that will work for a very small tissue, or the first few 100 microns at the edge of a larger tissue, but no more. Okay, so it's not going to get us close to that, 65 elephants organ scaling. Okay. And so very recently, we've been saying, well, can we use our laser sintering of sugar technologies coupled with some technologies that I had actually worked on as a postdoc when I was working with Chris Chen quite a while ago now. And the idea here is, again, you take your laser sintered sugar and then you cast a cellularized material around that sugar, and then you can inject a slurry of endothelial cells and collagen into this pattern network, okay? And we're calling this network that we inject with endothelialized network and collagen, we're calling them tracks. And I'm going to tell you why in a second, okay? These are not channels that are lined with endothelial cells. I want to really carefully separate that from what other folks have been doing in the field. I think the most common thing, right, is that you create networks like this and then you endothelialize the channels, the edges of the channels. That's not what we're doing here. We're instead injecting endothelial cells and collagen to form what we're calling tracks, okay, into these tissues. This is work that's been done by Liv Prado and Sharina Malkani in the lab. So here's what these tracks look like after they've been injected, okay? And so here you've got endothelial cells. We're labeling the endothelial cells, so that's what you see. And what we found, and this is again based on work from Chris Chen's lab way 15 years ago now, actually. And it's also very, even before that, based on a big literature in the cancer field, actually, that shows that cells will migrate along aligned collagen. Okay. And so what we find here is that after we inject cells and collagen into these tracks, is that the endothelial cells will actually then condense down around the collagen after just a couple of hours in vitro. Okay. So when you first inject the network with the cells, you'll get endothelial cells and your fibroblasts, right? And then over the course of time, those tracks or cords will contract down around the collagen, okay? If we then take a cross section of this, like here, okay? So you've got your endothelial cells contracting down around the collagen, which is here in yellow. And if we take a cross section of that, so here's a cross section of the gel with the different tracks. And if we take a zoom in here, a higher magnification window of this little tiny one, this is a stain for Sirius red, and that labels collagen, okay? And so what you see again is there's this sort of self-assembly of collagen within that track And then these little green things here are the endothelial cells, okay? So I hope you can see how the cells are kind of contracting it down into the center, okay? And this is an SEM image. Again, thanks to the reviewers for making us do this. You can see that each track is kind of poking out in this cross section, and you can see the collagen here, okay? And so these, again, are not endothelial cells lining the channel, but they're tracks that cells can migrate on, okay? So what happens when we implant these in vivo? Okay, so now we're taking one of the beauty actually of using a sacrificial templating approach is that you can cast virtually any material that can retain its shape around the sacrificial template before you flush it out, before you flush the template out, right? And so what we've done here is we've patterned a couple of different tissues, okay? So one is we've created blank tissue, so that's we take fibrin and we cast it around the sugar template, and then we flush out the sugar template, okay? And so we're just left with channels with nothing else, okay? We do the same thing within this case of 5% gelatin methacrylate, okay? And then we in our control conditions, we also then inject the tracks, okay? So this is again endothelial cells and collagen, which contract down and form these tracks, okay? And we do that in both fibrin and gelatin and JELMA. And what I hope you can see right off the bat is, yep, the tracks do seem to support. So this is the negative control without the tracks, okay, in both fibrin and JELMA. And then move down, here we've got the tissues with the tracks, both in fibrin and JELMA. And what I hope you can see is that there's quite a lot of endothelial cell self-assembly throughout the tissues, and that around those tracks in particular is where you start to see the most blood. And not only that, but there's quite interesting morphological differences between the fibrin and the JELMA cases, which I'll talk about a little bit more in a second. So if you take an InSen and zoom in, you'll see these self-assembled networks, right, that contain host blood in blue. And here's one in JELMA. This is near the inlet, so you can see the mouse vessels reaching in. And then you can see them sort of tracking down that collagen. Again, so these things work like railroad tracks down which cells can form vessels, okay? And one of the things that I think was neat about this data, again, is just the differences between the two very simple biomaterials. And again, these type of studies where they seem boring, and but like frankly, we need to be doing more of these studies where, you know, if we're going to have, for example, for acute liver failure, what we really want is a tissue that mostly retains its shape and that we can put in that works well and is really functional for the 2 weeks, and then we can take it back out again, right? So we want a tissue that doesn't become too entangled up with the host, that sort of retains its structure, and that has good perfusion throughout, right? And so one of the things we saw is, not surprisingly, right, for fibrin contracts down. This was its original shape going into the animal. This is the explanted shape coming back out. And not only that, but with the JELMA tissues, we found that we had better, the tracks had better, basically blood distribution across the entire network, as opposed to the fibrin, which interestingly had those really, really dark, a lot of blood, host blood out on the outside, but much less in the interior. So there are both self-assembly, compaction, and then overall structural differences, even with the same topologies, just encasing them in a different biomaterial, right? And so I should say the other thing that we found that was different, right, and this is probably partly just due to the fact that the fibrin tissues compact down so much over their time in the animal, is that in this case, when you do immunostaining for endothelial cells, if we look at the density of endothelial cells in the fibrin tissue, we have a higher density compared to the gelatin methacrylate tissues, right? And so one of the things we wondered is, if we simply, you know, in our first experiment here, what we did was we had these tracks, and then we had really sparsely seeded endothelial cells in between. So you still have a lot of empty space, right? We wondered if we just increased the cellular density in here in the interior, if we could then basically get the best of both worlds, where we can get infiltration throughout the whole tissue because of the tracks, but then we also get vascular self-assembly throughout the whole tissue because of the cell density. And so now after we've combined all of these things, what we're able to now get is these tissues that are, again, a couple of centimeters long, that are very densely vascularized throughout the whole tissue. And our hypothesis is that the DRACs are basically supporting vascular invasion down the tissues, and self-assembly. It's their chimeric vessels that are both human and mouse with our different immunostains so far. And then support self-assembly throughout the bulk, okay, of the tissues. When I first gave this slide, I was at a conference, a Keystone conference in Canada, actually, and Gordana had just finished speaking the day before, and one of the ways that she ended her talk was, she said, we design environments and cells build tissues across scales. And I just thought that that general notion, of course, she's talking about how she thinks about the field and all of the pioneering work that her lab has shown, right? And I just want to highlight that I really think we're seeing that same kind of thing here, right? So we can design the tracks, we can design what kind of biomaterial goes around the tracks, but in the end of the day, it's really the cells then that will architect the nuances of the tissues. So shout out to Gordana for being someone who has really put forth that idea into the field. So far I've showed you then that we can map tissues, we can start to get closer to building tissues. And I haven't talked at all about the society part that I mentioned earlier in the title of the talk, so I just want to spend a couple of more minutes there. I think that most of us in the bioengineering field think about our work, right? And we're spending most of our time down here, right? The work that's done in our labs, everything that I talked about today, was at either the cell level or the tissue level and somewhere in between, right? And I mean, we're trying to get closer to the organ level, but we're not there yet, right? And we really spend the vast majority of our time thinking about these spaces. And we say things like tissues are produced when communities of cells interact with each other and their environment, okay? Similar to what I just said about Gordana's quote. And one of the things that I would encourage all of us to do is to just pull our heads out of this level of scaling for just a minute to realize how important these other levels of scaling are in our profession, okay, and in our daily work, frankly, okay? So think about it like this: scientific ideas and progress are produced when communities of humans interact with each other and their environments, okay? And so basically, you know, what idea you deploy down here is really generated by the community of people that you've put together around you to get to that idea, right? And so it's really critical, I think, that we think more about these levels in our profession. And I would encourage you all to just think about that, even in your daily life, with respect to like, who am I eating lunch with, right? One example of this is we were fortunate to just, funny enough, I actually stole this slide from Adam Feinberg. And so the shameless plug thing is, you know, a little bit old from a different venue, but Adam Feinberg from Carnegie Mellon and I are currently co-leading a group of about 21 different labs from Carnegie Mellon, University of Washington, Mayo Clinic, and Berlin Institute of Health. And the idea here is to, again, bioprint human liver for within 5 years, within 3 years, large animals, and those studies will be done in years 4 and 5. And then with the idea being that after 5 years, we'll have data that would allow us to move into the clinic. Now, you know, I'm not sure that we'll actually achieve that very ambitious goal, but what will make it possible is the idea of bringing together 21 different labs all working on the same thing together, right? To get over some of those hurdles that we've talked about. So we're hiring right now, both Adam and I are, as well as the other labs. And so if anybody's interested in working on bioprinting liver for acute liver failure, please consider emailing us, okay? Another example of this is how do we build the communities that help make this work possible, right? And Gordana kind of alluded to this hub earlier when she introduced me, and I want to just highlight it to y'all because I hope that we're going to provide resources that will help you all in your daily work, okay? And so what this is, it's a virtual, we're calling it collaboratory, okay, that will put together resources for our community for biomaterials researchers, okay? It's a big group of PIs and advisory board, one of them is Gordana. And all of these PIs in our field are working together to give you guys all what you need in order to be successful, okay? And there's a couple of things that we're doing in order to make that possible. Number one is we're putting together resources in how do we actually translate our work? And this is not supposed to be the kind of translational resources that you get in every other setting. Okay, so right now, Karen Chrisman, Omid Visa, and Jason Burdick are working really hard to put together resources that will help you guys more easily translate your work. And so stay tuned. Those will be coming out really soon. There's also a group led by Stacy Finley that is working together, working to sort of bring computation more deeply into our field and helping you make that easier, make that leap easier. I think that many of us still remain a little bit intimidated by that leap. And then finally, a group led by Shelley Payton is helping to figure out how to disseminate these things and educate both our community and as well as the public about them, right? There's also going to be funding awards, which I'm going to talk about a second, in annual meetings, okay? And so first of all, if you want to get involved in this and just get access to all of these resources, all you have to do is email us to join. It's Humanity Unlocking Biomaterials. I know that's a really long email address, okay? Feel free to screenshot this. Email us to join, and you can you'll get added to a Slack network where you can get things like protocols and troubleshooting and, hey, do you have a cell population that does X, Y, and Z? You can ask anybody in the country or world, right, for those things that are on the Slack network. There's also going to be research grants in the Hub annual meeting, which we encourage you to come to. The research grants are $100,000 pilot grants that will be disseminated out to the community, okay? Year one, 6 awards, that's where we are right now, but we're going to be awarding up to 63 awards. So those of you that are PIs, stay tuned. Come join us at the annual meeting, which is coming up very soon. It's the day before the Society for Biomaterials annual meeting. Highly recommend coming. I'm going to dwell here again so you can screenshot it if you want to submit your applications. Last thing, second to the last, come to this Gordon conference that we're putting together with people in this field. It's going to be in May in Switzerland, so would love to have you join us for this. It's called Signal Transduction and Engineered ECM. And then finally, I want to highlight one community that I've been really impressed by within the last just like literally couple of days. And this is something that's been put together by Shelley Payton, who's the department chair at Tufts University, and then has a lot of leadership from actually Naveed Tavakal, who's the one that's doing a lot of the organization for these tissue talks, right? And so for anybody that's LGBT plus in BME, come join us on Slack for community. You can email Naveed, you can email Shelley, you can email me. And this is a great place to just interact with folks, share anything you want, and then also we'll have get-togethers and stuff too. So again, science is about the community of people. It's the context is everything, honey. It's about the what is surrounding both us as scientists as well as what's surrounding the cells that are doing the work when they're designing environments and building tissues. And I thought that at the very last conference I went to, Samira Musa has said this nice quote, right? Of all of the experiences that shape what I am and what I do, all of these things shape my science, right? And it's, the next one is going to be the most important. So thanks, everybody. Thanks to my lab. You guys are awesome. They've done all of the work and these collaborators, and I'm happy to take any questions, y'all.
Thank you so much, Kelly. This was fantastic. I think we can turn to Q&A, but you have very many questions, so we will need to ask you to please
I
answer.
talked forever. I'm
No,
sorry.
it's fine. It's fine. We really enjoyed your lecture, but then, like other speakers, it would be great if you can answer directly. We will provide you with the contact information. But
Love
let's take at least a few questions. And before we do that, I would just like to announce the next week's speaker. This is Mo Khalil from Boston University, and he will be speaking about synthetic reconstruction of complex cellular functions. So Q&A. Hayon Chen is asking, what are your thoughts about existing industry solutions, such as, for example, Satellite Bio? You probably know about this company. It's a liver
you.
regeneration company that are simply injecting hepatocytes infused into hydrogel into the body for systemic detoxification.
Yeah, so that's funny because Satellite Bio was founded by Sangeeta Bhatia, who, as y'all know, is fabulous. And some of that was similar to my postdoc work, right? And so like this is very similar to it's work that kind of led to this, right? And I think that that's really potentially powerful if, and I say if, the amount of function that can be provided for the patient is enough to treat whatever disease it is they decide to go after. Okay. So with the liver, it's really important that we think very specifically about what disease or what condition that the patient has that we're going to go after to treat, because in some cases, you only need to, treat or to recapitulate maybe just a couple of % of organ function, whereas in other cases, you need to recapture 100% of organ function, okay? And so it's very possible, my guess is that Satellite Bio will try and go to treat conditions that only need to recapitulate to or to replace a couple of % of organ function, right? In that case, that would be awesome. But the likelihood of treating something like acute liver failure with that technology with just a simple injection would be really tough. Okay, I'm never gonna say never, because, I can't say. Maybe they figured it out, but, time will tell. I wish them luck. I hope that they're successful.
So let's try one more question. It is also about the time scales. It's Francois Chesnay question. Self-assembly of capillaries takes several days. How do you envision avoiding hypoxia that happens within hours, so much, much faster, in 3D printed, large-scale, highly metabolic organs such as liver?
Yeah, I mean, that's a great question. I wonder about this myself, right? I really think that we're going to need to try both ways. I've seen first of all, one thing that has surprised me, frankly, is that this can work at all. So what we've seen is that despite the fact that it will take a couple of days for the vessels to self-assemble, there are some hepatocytes that survive and do well in the right settings, they'll remain functional, right? So it's possible that even with that hurdle, it will still work okay, right? That said, I agree with you. And ideally, what we would like to be able to do is get this to happen in vitro before we would put it into an animal and not really need to use the animal as the bioreactor as much. Unfortunately, the field's not there yet. We're trying to work on that ourselves, right? Using, various in vitro reactors that we're developing. And I think that time will tell whether we need to basically produce a whole vasculature before we put it into the animal or not, whether we can, inject a hydrogel and let the body do its thing. To be honest, I don't know the right answer right now. And because of that, my lab's working on both prongs, right?
I think it's very interesting what you say, thinking about importance of vascularization. It's uniformly important. But like if you talk about vascularization of the heart, it needs to happen at time zero
Yeah.
because cardiomyocytes that die, I mean, they don't proliferate, so you are left with whatever survived. If
Mhm.
it liver, it seems that you're saying that even very small fraction of the cells that survive can can start proliferating and then making up for the loss of
That's-
cells as the vasculature is coming in. I mean, time will tell, as you say, but it seems that liver has more options.
I agree. And actually funny that you say that, Gordana, because I started out my lab in heart and we've moved into liver, and it's partly because that's where we were funded, frankly. And then it's partly because I realized that it's got more options, right? And so, for what it's worth, I think that because of this interesting regenerative capacity of the liver, you can hijack and use that, right? So like you said, if you can get a couple of cells to survive, and then if you can get those remaining cells to regenerate and grow, they do have that capacity. I mean, of course, it's a matter of actually getting that to happen, right? In a controlled way that's safe and, but I think we're getting closer. We'll see.
Okay, so we can end on this very optimistic note, and then everyone who actually posted the question, you will get an answer from Kelly. We will take care of the logistics. Thank you, Kelly, again. This was really wonderful, very inspirational, and thanks everyone for attending and for asking very insightful questions. So see you next Wednesday.
Take care, y'all. Thanks for coming. Bye, Gordana. It's awesome to see you. Bye, Naveed. Thank you.